Great. Well, good morning, everybody. It's a real pleasure to take part in this course on genomics of dermatology. The title of my talk, Exploring Skin Cell Heterogeneity, is a topic that I've been working on literally since I was a postdoc. But it has entered a really exciting phase through an initiative that I'll tell you about called the Human Cell Atlas. So in this talk, I'm going to tell you about some of the heterogeneity that we're seeing in the epidermis and the dermis, and then give you a little bit of a flavor of the direction of travel for this area of research. So the Human Cell Atlas was launched officially in October 2016, and it's a global effort to map all of the cells in the human body. And already it's providing new insights into human physiology and disease mechanisms. And it's also driving new approaches to integrating different types of data to collaboration and data sharing. It'll come as no surprise to this audience that the skin features prominently in the Human Cell Atlas. And if you go to the website, you'll see that the network coordinators are Maria Kasper at the Karolinska Institute and me. So do contact us and help to increase the impact of this part of the Atlas. And I just want to acknowledge that in the early days of the project, the Chan Zuckerberg initiative were incredibly important, not only in providing priming funds, but also in bringing people together with very different sorts of expertise to imagine what a fully integrated atlas could look like. This diagram explaining the sorts of approaches that people take is actually taken from a review in a nephrology journal, hence the kidney in the top left-hand corner. But essentially the strategy is similar for the skin. So the starting point is generally a tissue biopsy. The cells are disaggregated, and then the RNA in individual cells is sequenced using a number of different potential platforms. There's then a lot of computational analysis required to make sense of the data. And of course, we then want to get spatial information about where the different cell types map into the intact tissue. Back in 2006, Kim Jensen, who's a professor in Copenhagen, he was my postdoc at the time, and he and I could see the potential of using single cell gene expression profiling to find new markers of epidermal stem cells. We published this paper in PNAS, and we were working, our starting material was cultured human keratinocytes. And I just show you this to show you that at the time, we were able to analyze just over 300 cells from the basal layer of the epidermis, and we got out of that 9 informative stem cells. It was a very successful project, and it led to the identification of LRIG1 as a regulator of stem cell properties, not just in the epidermis, but in many other epithelia. But if we fast forward now to the present, this is the Skin Cell Atlas collaboration, which is led by Sarah Teichmann at the Sanger Institute and Mazlifa Hanifa from Newcastle. And the team at King's, which is participating in this really interesting collaboration, is led by Magnus Lynch. So if you think about the number of cells that Kim and I were working with back in 2004, you can see that in this slide, through Muz's efforts, the number of cells that we're looking at has passed 500,000. And I think, in fact, now we're up at nearer 600,000. Instead of focusing on cells that we've cultured from human epidermis, we've got cells from epidermis and dermis. And we've also got samples from healthy adult skin from patients with atopic dermatitis and psoriasis. And Muz has been working with the developmental tissue bank in Newcastle to also obtain data from fetal skin. So the amount of information that we have at our disposal is really massive and incredibly exciting. So what can we do with this information? Well, of course, I'll tell you first about the epidermis, because that is my first love. And I started working on the epidermis when I was a postdoc with the late Howard Green. And he had shown back in the 1970s that it was possible to isolate cells from human epidermis, grow them in culture, and they would make multi-layered sheets of epidermis, which only 6 years later he showed could be used for grafting. So from these early studies, we know that stem cells exist in the epidermis and can be expanded in culture and used as a stem cell therapy. But it's interesting to note that the assay that Howard used for quantifying stem cells was to count the number of colonies growing in the dish. And this is an assay that we use in my lab and many other labs routinely to the present day. This is just a picture of a colony forming assay that we carried out when we wanted to measure or evaluate different types of serum for growing the cells in. So using this in vitro model of human epidermis, a lot of us have worked hard to understand the nature of stem cells and also the pathways that regulate the onset of differentiation. And in recent years, my lab has focused not so much on the stem cells, but on the early steps by which a cell leaves the stem cell compartment and makes the decision to move into the suprabasal layers and differentiate. And so this transition point is something that we call commitment. And a lot of work, in particular led by Ajay Mishra, a former postdoc, and now by Toru Hiratsuka, a current postdoc in the lab, has led to this very dynamic model of what regulates the stem cell compartment. So we envisage a key pathway in regulating exit from the stem cell compartment as being the ERK MAP kinase signaling cascade. And in this schematic, you can see how we believe the system is working. We're using involucrum, which is a spinous layer marker, as our differentiation marker. And we have a fluorescent reporter, which tells us which cells in real time have started to differentiate. So we start over here with the stem cells, which have got high levels of ERK activity and don't express involucrin. And we know that the differentiating cells, which have left the basal layer, have low ERK levels and are involucrin positive. And what we found is that this transition or commitment phase maps to periods of changes in both ERK levels and pulsatile activity. And these changes are regulated by protein phosphatases, one of which, DUSP6, modulates these pulses, which you can see as spikes here, and another one, DUSP10, modulates overall ERK levels. It's hard to imagine in many ways how this sort of detailed molecular biology, highly advanced imaging, and a lot of mathematical modeling could map onto a reference map of cells in the human cell atlas. But of course, scientists have a tendency to get so attached to their models that they're blinkered to reality. And we were very, very keen indeed to find out whether or not this hard work that we'd done in vitro was indeed relevant to the data coming out of the cell atlas. So two specific predictions from our work were the existence of multiple populations of keratinocytes in the basal layer. We've previously hypothesized that there might be two different populations of stem cells in addition to the committed cells, and also the prediction that transient upregulation of protein phosphatases like DUSP6 and DUSP10 plays a role in transitioning into the suprabasal layers. So to our model, we apply the might of the data set that we have through our collaboration, and Victor Negri, a former postdoc in the lab, who has recently moved to AstraZeneca. So what Victor was able to do in collaboration with Muzz and Sarah and the rest of the team was extract from the big data set single cell data that only pertained to healthy human interfollicular epidermis. And he looked computationally for the existence of different subpopulations of cells, which are represented here in what's called a UMAP plot. And because we know a lot of the markers of the basal layer and the different suprabasal layers in the epidermis, we could assign some of the populations with considerable confidence. For example, keratin 14 is expressed in the basal layer, and in healthy epidermis, keratin 10 is expressed above the basal layer. So just looking at this plot, you can see that already in the UMAP plot, we predict the existence of 3 distinct populations of basal cells. You can see also that we can pull out different populations of cells in the spinous layers. Now, in terms of the granular layer, you can see a very small number of cells here. And this highlights something which is a caveat, not just for our work, but for all human cell atlas work. And that's to say that just because you have a low representation of granular cells in the map, that represents their true abundance in vivo. And of course, if you've ever tried to disaggregate epidermis with enzymes, you'll know that it's very hard to disaggregate the cornified layers, which don't appear on this plot as well, and to get good representation of the granular layer. But nevertheless, this looks interesting. And what particularly caught our attention was the predicted existence of these 2 populations here, which Victor has named transition 1 and 2. So essentially, the idea is that you have different basal populations, you have a transition or 2 transition populations, and then you move into the spinous and granular layers. So what is going on in there? Well, of course, we wanted to know if DUSP6 and DUSP10 appeared on the UMAP plot, and if so, where. And you can see I've shown the 2 transition populations circled here, and DUSP6 and DUSP10 do indeed map where we would predict. What's even more pleasing about the data is that in experiments that we'd done in vitro, we had predicted that as you push cells from the stem cell compartment where p63 is expressed into the differentiation compartment, there's transient upregulation of DUSP6 and DUSP10, but the upregulation of DUSP10 lasts longer than for DUSP6. And that's completely consistent with the wider spread of DUSP10 than DUSP6 that you can see here. Another thing that Victor has done, and this is very preliminary work, is to look at how the different populations of cells that we'd found in healthy skin map to lesional and non-lesional skin from patients with eczema and psoriasis. Here, actually, my prediction had been that there would be an abnormal expansion of those 2 transition populations, which are now shown here. But in fact, we didn't see that. We saw that their abundance in the map was shrunk, as was the abundance of the different basal populations. And instead, what you see is an expansion of the spinous layer populations. So what's nice about this is that it gives us some predictions that we can go back to test mechanistically in our experimental models. So to summarize this part of my talk, we had 2 predictions from our studies in vitro with human epidermis, and both of those are supported by the Human Cell Atlas work, in addition to giving us pointers for further mechanistic experiments that we should carry out. And I suppose you could imagine that armed with the Human Cell Atlas, if you're trying to navigate through a tissue, you can use it like a Google map to help tell you where you are and areas that you should explore. Now, for the last part of my talk, I would like to turn to the second love of my life, which is the dermis. And here, the challenge has been a bit different, because many of you will know from the pioneering work of my former postdoc, Ryan Driscoll, we were able in mouse models to provide convincing evidence that dermal fibroblasts are not all of the same, but they represent distinct lineages, and also they have distinct functions. I'm not going to take you through this in any detail because the work is published, but essentially the question was, do different subpopulations of fibroblasts exist in human as well? Are they functionally distinct? And can we use the markers that work in neonatal mouse backskin as a guide to studying human fibroblasts? So the predictions from our studies in mouse and from important pilot studies that were carried out by Magnus Lynch and Christina Philippius, we predict that there should be multiple populations of dermal fibroblasts with distinct functions in adult human skin. And what we're looking for more is evolutionary conservation of particular signaling pathways in the different subpopulations, rather than specific markers that we can use for flow sorting of the cells. Magnus and Christina published a paper back in 2018, which had more cells in it by far than the work that Kim Jensen and I had done before. But nevertheless, the number of cells that we looked at was quite small. But you can see from this, these are T-SNE plots of single-cell RNA-seq data looking at different markers. So we could pick up different populations of cells, and as we've published, we could see that the expression of some of the populations was spatially segregated. In particular, you can see that collagen 6A5 is tightly confined to the uppermost papillary dermis, and CD36 is abundant in the reticular dermis and also in the adipocytes. In skin reconstitution assays, Christina obtained convincing evidence that these different populations of cells are functionally distinct. But what happens if we go now into the bigger map that we have through our collaboration with Muzz and Sarah? Actually, I have to say that this is the work of Clarisse Garnier. And in this initial study, she's not taken the work from our current data set, but she's merged the data that from two other groups that was published to Beebe et al. In 2018 and Soli Boldo in 2020. But essentially, it's the same principle as we have with the new data sets, that you can use a UMAP plot to ask where are the keratinocytes, where are the vascular cells, T cells, and fibroblasts. And if we look in here at the fibroblasts, sure enough, as predicted, there are distinct populations of cells. We're quite pleased because with our smaller data set, we had predicted there would be about 5 subpopulations, and indeed that comes out to be true. And this, of course, is a rich source of new markers that we can use to interrogate fibroblast function. So the work on the fibroblasts confirms the existence of different populations of cells. It gives us new markers that we can use to map those cells. And going back to the issue of signaling pathways that control dermal properties, it gives us some really interesting candidate mediators that could explain mechanistically the variation in function of the fibroblasts. Now, I'm almost at the end of my presentation, but I want to give you a taster of where we're going next. And this, to me, in many ways, is the most exciting part of the project. Wouldn't it be great if in the clinic you could non-invasively look at a skin specimen and diagnose the changes in its cellular composition without having to take a biopsy? I don't know if that will ever be the case, but it is a challenge that we're really interested in the lab at present. And the approach that we've taken is to use a technique called optical coherence tomography, equipment called VivoSight. And I'm sure many of you will have seen this. You essentially putting an ultrasound probe over the surface of the skin, and you can very easily map out the different thicknesses of epidermis and dermis. You can pick out the basement membrane. You can use 3D reconstruction to get information about the skin vasculature, and you can get some very interesting maps of regional variation in the thickness of the epidermis, which is shown here. So what Clarisse has been doing is to have a look at the OCT images of the skin vasculature. And she's been able to image the superficial vasculature and the deep vascular plexus, but we haven't in these images attempted to model the vasculature in the subcutaneous fat. And this just is a sample of what she's done, looking at vascular density in different body sites of healthy adult volunteers. And just by eye, you can see that it would appear that some parts of the body have a much higher vascular density and complexity than others. In particular, it won't surprise you to see that the lip is particularly well vascularized if you compare that with the forearm and the abdomen. So can we map this to changes in different cell types? Well, the answer is, honestly, we don't know. But we do know that the histology of the skin in different parts of the face differs, as does the 3D organization of the vasculature. And what we're looking at currently is whether we can pick up different subpopulations of endothelial cells and pericytes, as well as fibroblasts, and correlate changes in abundance of these different populations of cells with the different vasculature architecture that we see. So that, of course, is work ongoing. But of course, it may be that the vasculature does not depend on the composition of the cells. It may depend on signaling pathways. And of course, it could depend on those different populations of fibroblasts. At this stage, we just don't know. So that's the end of my talk. I'd just like to reiterate how exciting the field of skin cell heterogeneity is now. It gives us a reality check for our experimental models. It's giving us new insights into skin physiology and disease. And very importantly, it's driving collaboration, data integration, and data sharing in a way that I've certainly not seen before. And it is very healthy for our field. Now, it seems a long time ago, but last Christmas we were celebrating in my house, no face masks, no social distancing. I'd like to say a huge thank you to everybody in my lab who has contributed to this work. They are an amazing team. So thank you very much and goodbye.